And it is now time for the next slammer, the fourth slammer of tonight, and this will be Agata Mossakowski. She's a postdoc at the University of California, Davis, and she's going to talk about treating muscle diseases. Agata, the floor is yours.
So by now, you know by heart how many people out of 10,000 still sleep with a stuffed animal. And you know also that 2,300 prefer Pepsi over Coke, and that 800 out of 10,000 adults say Wi-Fi instead of Wi-Fi. And we know that's rare, but only 6 out of 10,000 have a muscle disease. Now, I've already told you now that muscle diseases are rare. They are hard to diagnose and pretty much untreatable. And so the problem is that I'm talking about muscle diseases in general, although there's hundreds of different muscle diseases. Some of them are induced by virus, some triggered by medication, quite a lot of others caused by autoimmune disease. But then there's a big and very significant part of them that are caused by genetic mutations. And at this point, let me introduce you to Ramon. Ramon was born in Chile, and he had a very normal childhood in his hometown of Santiago, grew up as a healthy teenager. But then when he was 35, he noticed that some things were quite off, and he noticed first little things like tripping, being exhausted, but later his muscles became so weak and thin that he had to use a wheelchair. And once doctors found out what was actually wrong with Ramon, they couldn't do anything because Ramon's disease is so rare that we barely know anything about it. See, Ramon was born with a mutation in his Desmond gene. His Desmond gene is responsible for making the Desmond protein, which is a very important part of the cell and is pictured in yellow in this picture. And it's a part of the muscle's skeleton, the cytoskeleton, and it provides stability and structure to the cell. So to find out how the problem in Ramon's cytoskeleton causes his disease, we made Ramon into a rat. First, we saw that Ramon rats weren't that different from healthy rats, so we started exercising them. And that's where, quite literally, things began to go downhill for the Ramon rats. They couldn't keep up. Not only were they unable to run for long distances, but they also got worse and worse each and every day. And when we looked at the muscles under a microscope, we saw that the muscles broke down. And we also saw clumps. Clumps, I hear you ask? Yep, clumps. And this is where it gets really interesting. You see, with Ramon's mutation, the Desmond doesn't just go away. Instead, the Desmond in Ramon's mutation gets misassembled or becomes misfolded, and that causes two things. First, it loses its function as a skeleton, and the muscle cell becomes, well, weak and squishy. But second, it also doesn't get recycled properly anymore. Usually there are recycling mechanisms in the cell that repair and reuse part of the proteins and build them into new proteins. But when there's too much of the misfolded Desmond, the recycling machinery gets overwhelmed, doesn't work for other proteins as well. They form these clumps, and that's where the muscle can't repair itself and breaks down. So what can we do? Well, first of all, we will have to overthink how we prescribe physical therapy to our Desmond patients, because if treadmill running causes that much harm to our Ramon rats, maybe we should be aware of how much physical therapy our patients do and how that affects them. But second, we have also some other ideas. Because interestingly, the proteins that are at the center of Alzheimer's disease are quite similar to our clumps. And other scientists have seen some pretty cool results in detangling those Alzheimer clumps with a compound called edaravone. And that's what we started to use on our Ramon rats now. We had some pretty exciting first results, and the future will show how we can use it in patients one day, because in the end, we want to use the knowledge that we gained in our Ramon rats to help Ramon himself. So to sum it up, 3 things. First of all, I hope you've learned that there's a protein called Desmond, and even if you've heard about it for the first time today, it's an important part of the muscle skeleton. Second, animal models can be really useful to study rare diseases. And third, muscle diseases are rare, but that does not mean they are unimportant. Thank you very much for your attention and patience.